So just to review what we talked about in the previous video, so you've got 46 chromosomes in the beginning, then DNA replication happens, that gives you 92 chromosomes. It divides, which gives 46 and 46, then no replication happens, and you end up with 4 cells that have 23 chromosomes. Now, this is going to be important when we talk about egg and sperm, okay? That's the products of meiosis. That's the whole point of meiosis, is to make these things. So what's going to happen is you've got a sperm with 23 chromosomes and you have an egg with 23 chromosomes. Those are going to come together and make a zygote that has the 46 chromosomes. So that's how we as beings have 46 chromosomes. So I just wanted to go over that just to make sure that that kind of made sense. All right, let's talk about the process of meiosis. So there's going to be meiosis 1 and meiosis 2. So right now we're going to talk about meiosis 1. So look at the names of the phases. Hopefully that sounds familiar to you. If not, go look at the mitosis videos because you got to make sure that you understand those. Okay, so prophase 1. If you remember in mitosis, prophase is going to be where the chromosomes become visible, nuclear envelope starts to dissolve. Similar stuff is going on here, but don't forget we have that crossing over happening. All right, so the DNA coil's tighter, chromosomes become visible, and what's going to happen is homologous chromosomes are going to associate with each other with something called synapsis. And that's basically where the homologous chromosomes are going to find each other and match up. They're going to do that because they want to exchange genetic information. So in the picture I have on your PowerPoint, it's not like some superb picture, but here you can see you've got the chromosomes lining up with one another, and what's going to happen is they're going to exchange genetic information, and it's a perfect exchange, right? So you don't have somebody like taking the hair color one and also taking the eye color but not giving any over. It's always going to be a direct exchange. If you look at these guys at the end of prophase 1 and beginning of metaphase 1, you can see that this little blue guy has a little chunk of red, right? This little blue guy has a chunk of red. This little red has a chunk of blue. So the crossing over has happened, right? All right, so if we go back to the notes, synapsis is going to be where they come and join up together. Now, what's going to happen is crossing over, and they're going to exchange DNA segments. Then, when crossover is done, what's going to happen is, I should mention that there are going to be these little cross bridges where this has happened, and that's called the chiasma or chiasmata. And so chiasmata is just going to be plural. And so what's going to happen is those are going to be little bridges where you can actually see that crossing over has happened. And I think I've got a picture of it here. Yeah. So you can see a chiasma right there where crossing over has happened. What's going to happen is as it gets closer to the end of prophase 1, those chiasma are going to move towards the ends of the chromosomes. All right. So what's going to happen is they're going to try to pull away from each other at the end, right, just to kind of get set up. But what's going to happen is they're going to be held together by a couple of different forces. First, you've got 2 sister chromatids that are held together. Remember, those are identical copies of a chromosome. And then the homologous chromosomes, chromosome 2 from your mom, chromosome 2 from your dad, those are going to actually be held together where those chiasma are going to be. Oops. Moving down. Okay, I lost where I was. Okay. So the chiasma, like I said, is going to be that little bridge where the DNA segments have exchanged. And that's pretty much it for prophase 1. So the really important thing you want to get out of prophase 1 is that the crossing over happens. Now in metaphase 1, I'm going to let you take a look and see if you notice anything different about metaphase 1. Yeah, that'll work. So it's kind of on its side, so don't think about that. But if this was turned the way that we're used to drawing it in class, do you notice anything weird about the way that they're aligned on the metaphase plate? Think about how it looked in mitosis, and if I get my little drawing board out, I can kind of show you, if I can get it to come up. Okay, so I'll draw over here. Whoops. Oh boy. I'm telling you, sometimes technology and Fleur don't get along. Okay, so I'm going to write over here. Wow. Just doesn't like me today. Okay. Do you want to work? Yes? Okay. So I'm going to write over here mitosis, and over here meiosis. So when we get to metaphase in mitosis, remember when we drew it, it looked like that, right? In meiosis, what's going to happen is it's going to look like that. So the difference is that homologous chromosomes are lined up next to each other instead of in this long line, and if I wanted to make it perfect, I would have put all of them in there, right? So that's going to be a little bit different from what we're used to in mitosis. Not a huge difference, but what's going to happen is, and I'll change colors for this, when you have those microtubules come out and attach to these guys, it's going to be a little bit different from mitosis, right? So, oh wow, now I'm really glad that I drew all of these million chromosomes. Okay, now, in mitosis, what's going to happen is when these red microtubules get shorter, that's going to split the sister chromatids apart. In this situation, the microtubules are only attached to one side of this, so this entire thing is going over there. And this entire thing is going over there, right? So that's another difference. Okay, so that's going to be something that we want to notice about metaphase 1. All right, so the pairs of homologous chromosomes align down the plate. Chiasmata are helping them keep them together so you don't have like weird mismatched ones. And the microtubules are going to attach to just one side of those centromeres, which I showed you in the picture. Okay, so those are going to be the two phases that are really, really, really different as far as mitosis and meiosis go. Then we have anaphase 1, and what's going to happen is very similar. You've got the kinetochore microtubules are going to get shorter, and that's going to pull entire sets of homologous chromosomes over to one side of the cell and the other. Sister chromatids are not splitting, so the X is staying together and going over here or over here, not like in mitosis where the X splits apart, right? Then we have something to talk about here, independent assortment. So one thing about meiosis that we talk about a lot is the fact that there's so many chances for these end cells to be so genetically different from one another. And so if you look here, you can see that thanks to crossing over, all of these chromosomes are now a little bit different from one another. Now, when we have metaphase happening, there's a lot of possibilities of how they could align themselves on the metaphase plate. You could have all the paternal ones here and all the maternal ones here, or you could have it mismatched, right? As long as you have like chromosome 1 next to chromosome 1, chromosome 2 next to chromosome 2, it doesn't matter what side the mom or the dad ones are on. So there's another way that you can end up with a lot of more genetic differences between these cells at the end. Okay. Then we have telophase 1, and what's going to happen at the end of telophase 1 is the nuclear envelope reforms, and it's going to eventually split into two cells, right? So going back to our PowerPoint, sorry, allergies. Okay, we've got prophase 1, metaphase 1. Here's anaphase 1, entire sets of chromosomes are going to opposite sides. Telophase 1 in cytokinesis, the nuclear envelope is reforming. Now if you look, they do look genetically different from one another, right? Then we're going to get into, this is the whole thing together, meiosis 2. In meiosis 2, you're going to notice this is exactly the same as mitosis. So in prophase 2, nuclear envelope dissolves again. Metaphase 2, they all line up the way we're used to down that metaphase plate. Anaphase 2, now the X's are actually being split apart. And then you have telophase 2 in cytokinesis where it splits into those 4 cells. So that is going to be just like what we learned for mitosis, right? Okay, so that's going to be pretty much the same thing. However, a couple of differences. No two cells at the end are going to be alike due to that crossing over and independent assortment. And we'll get into this other stuff in the next video, but I just wanted to show you kind of the phases of how that kind of goes through.